So thank you very much for this great honour and great surprise. I was nominated by one of my colleagues at Heriot-Watt University's Dr. Stuart McGregor for this, and so I have him to thank for that nomination, and a lot of people to thank at the end of the presentation who've helped along the way as well. So I'm going to talk today about the toxicology of nanomaterials and looking back and into the future to look at where we need to go, what's the best way forward in terms of understanding nanomaterial toxicology. So historically, I came into this area about 20 years ago when there was a lot of epidemiology data to suggest that elevated particulate air pollution was associated with increases in ill health, but also deaths in the population. And that epidemiology seemed to suggest there were respiratory effects associated with particles and cardiovascular effects. And those effects were acute, short-term effects within a few hours or days of a pollution episode. And more recently, that epidemiology data has demonstrated long-term effects as well. So things like increased incidence of cancer, developmental effects, and effects on the brain. And there's lots of studies from all around the world to demonstrate these epidemiological observations relating air pollution to ill health. And for example, this is an intervention study. It's in Dublin. They used to burn lots of coal in Dublin for commercial and domestic fuel purposes, and that was associated with high levels of particulate, black smoke pollution, and gases such as sulfur dioxide. They banned the burning of coal in this environment, in this city, and they saw an immediate drop in the pollution levels and an immediate drop in the death rate due to cardiovascular and respiratory causes. So there's widespread epidemiology showing that pollution and particles in particular can cause adverse health effects in humans. So particulate air pollution is monitored in our towns and cities as PM10. 10 represents 10 micrometers. These are the largest particles that you can inhale into your respiratory system. But actually, if you look at PM10, the composition of particles is very diverse. They come from lots of different sources. They're chemically diverse. They're diverse in their physical properties. And that can vary over time and from place to place. And so the key is to try and identify which of those components within PM10 are responsible for driving adverse health effects. So in the mid-1990s, a group in Edinburgh with Bill McNee, Ken Donaldson and Anthony Seaton put together a hypothesis which they called the ultrafine particle hypothesis. And they suggested that the smaller particles in the nanometer size range within air pollution were responsible for driving these adverse health effects. And that hypothesis was based on an observation published from Rochester with Gunter Oberdorster's research group. And they had observed that small or ultrafine size range titanium dioxide particles caused a response in the lung of animals which they didn't observe with larger particles. And this response involved an activation of the immune system known as inflammation. So they measured inflammation in the lung tissue of these animals, and they found that the 21 nanometer particles induced an inflammatory response. When they stopped the exposure, there was some recovery, but it remained sustained for a number of weeks after the exposure, whereas the larger particles didn't. So Bill and Ken and Anthony and others used that information to generate the ultrafine particle hypothesis. And they were focusing on the respiratory system and looking at how these particles might interact with the airways, but also the deeper lung where the gas exchange occurs, such as the alveolar regions of the lung. Now, these different parts of the lung are well protected. We've evolved over history to deal with pathogens such as viruses and bacteria in the environment, and pollution, for example, from wood burning and from volcanic sources and wind-blown dust. So our upper airways are lined by a series of epithelial cells with hair-like cilia on their surface bathed in a sticky mucus. So if particles land in these airways, they're quickly cleared by the mucus that moves up and out of the respiratory system. If the particles deposit in the alveolar region of the lung, here the lung is protected by immune cells, such as the macrophage shown here. These can migrate from blood vessels into the air spaces, identify the foreign particle or pathogen, and ingest them and either break it down if it's digestible, such as a bacterium, or if it's not, so it's an inorganic particle, it can physically carry it from the lung, either with the mucus or by moving into the lymphatic system, therefore keeping the surface of the lung clear. And that's why we don't all die during a pollution episode or every time we get a cold. But sometimes this process goes wrong. Some people are more susceptible than others, and some particles are more toxic than others. So that system doesn't always work perfectly. So the ultrafine particle hypothesis was developed to try and explain why ultrafine particles might be more toxic in the lung than larger particles. And it seems that these particles, or it was hypothesized that their very small size and their large surface area, enabled them to generate reactive oxygen species, or ROS. These are highly reactive substances that can damage proteins, lipids, DNA, and cause damage to the surrounding cells. Actually, at low doses, rather than killing cells, they activate the cells. And they activate the cells to generate these proteins called cytokines. And these cytokines stimulate cells from the immune system, like the macrophages shown here, and other white blood cells such as neutrophils. And they stimulate them to migrate into the lung tissue to find the foreign particle and try and clear it. But as they do, these cells themselves also generate reactive oxygen species. So you now have a scenario where you have particles generating these reactive oxygen species as well as the cells. And in a normal healthy individual, they can probably recover from that. But somebody with a pre-existing disease that involves activation of the immune system or inflammation, this can help to make their disease symptoms worse. So this might be somebody with asthma or bronchitis, or somebody with cardiovascular disease or diabetes. Their symptoms could be made worse by this kind of exposure. So the group in Edinburgh took some PM10 particles collected from the centre of the city, and they instilled them into the rat lung, and they measured the amount of inflammation in the rat lung, and they found that it did induce inflammation or an activation of these immune cells. Then they took carbon particles from a commercial source, from Degussa, of different sizes. And they found that the 14 nanometer particles also induced an inflammatory response, whereas the larger particles didn't. So this started to support their hypothesis that the ability of particles to promote inflammation in the lung is related to their size. At this point, they wrote a grant application to the Colt Foundation. I'm glad to say Jackie Douglas is here with us today. And they funded probably one of the world's first projects to investigate the toxicity of ultrafine particles. And they continued to fund this work for well over 10 to 15 years with different people along the way, which provided some of the key data to look at how these nanomaterials or ultrafine particles behaved in the body. So the project that I moved to Edinburgh to work on in 1996, we wanted to test this ultrafine particle hypothesis, but it was really difficult to get environmental particles of different size fractions. So instead, we used commercially available particles of different sizes and different compositions to test the ultrafine hypothesis. So we had carbon black particles, we had polystyrene beads, we had titanium dioxide particles. And we started to look at the ability of these reactive oxygen species on the particles or associated with the particles to cause damage to the cells or to activate the cells. So one way to do this is to measure the level of oxidative stress in the cells. So the reactive oxygen species cause this oxidative stress. And you can do this by measuring glutathione, which is an antioxidant in cells. So these antioxidants protect cells against reactive oxygen species and become depleted if the reactive oxygen species levels go up too high. And we found that the 14 nanometer particles did induce this oxidative stress, whereas the 260 nanometer particles didn't. We could measure these reactive oxygen species using a variety of assays. For example, a fluorescent assay here, which was used to measure reactive oxygen species production by the 14 nanometer particles, but not by the larger particles. And we also demonstrated this for other particle types, such as polystyrene beads. And this is now widely used in the literature to compare the relative ability of different nanomaterials to generate reactive oxygen species. We then wanted to look to see what the impact of these reactive oxygen species might be on the cells. So we started to focus on the immune cells, the macrophages that are responsible for clearing the particles from the lung surface. And we measured their calcium concentration inside the cells, an indicator of their level of activation. And we found at sublethal doses, so doses that weren't killing the cells, there was a switching on of the calcium signaling mechanisms inside these cells, and that this calcium signaling was then responsible for activating proteins in the cell that drive inflammation. For example, this is a transcription factor called nuclear factor kappa B here, which is normally found in the cytoplasm of the cell, as shown by the green staining around the cell body. But when treated with a carbon black nanoparticles, it moves into the nucleus, so you now have a nuclear stain associated with this transcription factor. So from this, we were able to start to put together a mechanistic pathway, or an adverse outcome pathway, as they're now called, to try and explain how these particles might impact on the cells. So we have evidence that reactive oxygen species caused calcium signaling in the cells and activation of transcription factors that then switch on the genes that drive the production of the cytokine proteins that control inflammation. So this was all published in the early 1990s and 2000s, early 2000s, using the term ultrafine particles. But then, in around 2004, there was great interest in nanomaterials, mainly driven by a number of reports, such as a report from the Royal Society and the Royal Academy of Engineering, and by others, that started to look at nanomaterials and how they were being used in a wide variety of different applications and whether there were any risks associated with them. And these nanomaterials turned out to be the same size range as the ultrafine particles we'd been investigating in relation to air pollution, around 100 nanometers or smaller, so around the same size as macromolecules like DNA that make up cells. So there have been a number of definitions published over the last 10 years to define what a nanomaterial might be like. Most of them suggest that the nanoscale is between 1 and 100 nanometers, although for nanomedicines that can sometimes be larger. A nano-object has at least one dimension in the nanoscale, so that would be something like a disk or a graphene sheet, shown here. Whereas a nanomaterial has about 2 dimensions in the nanoscale, so something like a fiber or a carbon nanotube. And then a nanoparticle has 3 dimensions in the nanoscale, so something like a sphere or a cube. And there are more precise definitions than that available from the European Commission, from the British standards industry, but they more or less fall into those categories. So why did the industry and researchers become interested in man-made or engineered nanomaterials? But it appears that at the nanoscale, substances change their physical and chemical characteristics. So gold, instead of being a yellow, shiny metal, becomes a coloured material, pink, purple, blue, depending on its size, and it becomes much more reactive. You can start to use it for different applications. These are gold nanoparticles that have been imaged by near-infrared imaging. You can see them inside an animal. These have labelled a tumour inside a mouse. And then you can use their near-infrared excitation and emission properties to actually kill the cell that they have accumulated in. So you go from an inert material to a material that can be used to kill cancer cells. In fact, nanomaterials come in a very wide diversity of chemical characteristics and physical characteristics, from a simple carbon-based material such as carbon nanotubes and fullerenes, through to substances which have multiple different elements within the same nanomaterial. Things like quantum dots with cadmium telluride and celluride in the core, zinc sulfide shell and maybe a polymer coating, or liposomes used for nanomedical applications with a drug in the core, a lipid sphere, and then various different coatings on the surface to target them to a specific location. So when you start to think about the diversity of nanomaterials and their physical and chemical properties, this raises a number of interesting questions when you think about how they behave in a biological environment. So as toxicologists, we have a wide selection of nanomaterials to consider. But it's not just their variety in size and shape and charge and composition that's important. They're also then incorporated into a wide variety of matrices. They're in paints, they're in inks, they're in clothing, electronics, cosmetics, food packaging. Almost any kind of matrices you can imagine could incorporate nanomaterials. And they don't stay the same over time. You have the pristine nanomaterial made in the factory, you have it incorporated into the product, it ages, it's weathered, it may be released. So how do those different nanomaterials at their different life cycle stages behave? And this is important because there is likely exposure for nanomaterials for both humans and for the environment. There's purposeful exposure. For example, we apply cosmetics and sunscreen onto our skin. We put nanomedicines into our body. But there's incidental exposure where people working in an occupational setting might be exposed to nanomaterials, or consumers when using a product. For the environment, there are purposeful release of nanomaterials in some countries into the environment for remediation purposes, so clearing up contaminated land. At the UK at the moment, that's not allowed, but it is allowed in some other countries. And then there's also incidental release, for example, in wastewater. And again, we need to consider not just the pristine materials, but these different life cycle stages as well. When we've considered air pollution particles in the past, the respiratory or lung system has been the main route of exposure. And there have been some interesting studies by Wolfgang Kreiling and others in Germany that have shown that when nanomaterials are inhaled into the respiratory system, a small proportion, about 1% of those inhaled particles, can cross the lung barrier and gain access to the cardiovascular system. And they do distribute around the body to different organs. You find them, for example, accumulating in the liver, but you also find evidence of them in fetal tissue for pregnant animals, and you even find them accumulating in the brain. Now we also need to consider exposure via the gastrointestinal tract, because nanomaterials are incorporated into food, they're in food packaging. When we clear particles from the respiratory system, the mucus is swallowed into the stomach. So there are multiple routes of nanomaterial ingestion. And we also need to think about nanomaterials that are directly injected or implanted into the body. And as I've said, nanomaterials are available in a very wide variety of physical and chemical properties. And here are some of the physical and chemical properties that need to be considered when you think about their potential hazard. I'm going to talk in more detail about size, surface area and shape in a moment, but composition is key. If there's a particularly toxic component within the nanomaterial, you can expect that to contribute to its toxicity. Charge is important. Positively charged particles seem to enter cells quite avidly compared to neutrally charged particles. Crystal structure is key for some materials. Amorphous silica is a benign material, whereas crystalline silica is a carcinogen, for example. Solubility is important. If a particle is soluble, it's not going to be biopersistent. But if it contains toxic metals or toxic materials that can be released due to that solubility, then that can contribute to their toxicity. Strength is important, especially for fibers. Fibers which are brittle can break down, and this can influence their ability to be cleared from the body. This slide was put together in 2007, and then I used the term electrical conductance. We thought that electrical conductance might be important in determining toxicity, and in fact, a group in California with Andre Nell have shown that the ability of electrons to move within metal and metal oxide crystal structures, known as a band gap, is important in determining their toxicity. So there's a whole host of physical and chemical characteristics that need to be considered, not just size. So this was some work conducted several years ago by one of our PhD students, Martin Clift, using the macrophage cells, the immune cells, staining them with red to show the cytoskeleton or the cell body, and the green are fluorescent polystyrene beads of different sizes. And he showed that the nanoparticles and larger particles are taken up into these cells. but it's more extensive and it's quicker for the nanoparticles. This is important because we've got the same mass dose being added to these cells, but there's much more uptake for the nano-sized particle than for the larger particle, meaning there's a greater dose to these cells in these culture conditions in vitro. If you then think about this in terms of different dose metrics such as surface area, you can see that for a 100 micrometer particle and a 10 nanometer particle, the surface area dose in meters squared per gram is much, much greater. You're talking about a tennis court versus a piece of A4 paper in terms of difference in surface area. And then the percentage of atoms at the surface is also very different. You've got 0.001% here and 10% there. So there are lots of atoms available at the surface and available to interact with the biological matrices around them. So some work conducted by one of our PhD students at the time, Roger Duffin, who's now at Edinburgh University, showed that there was a good straight line relationship between the surface area dose of low solubility, low toxicity materials and their ability to induce inflammation. So this includes things like carbon, titanium dioxide, and polystyrene beads. But then if you took a particle, which is not a nanoparticle, but a highly reactive quartz particle, alpha quartz, which is crystalline silica, which is a carcinogen, it doesn't sit on the same line. And this is a carcinogen because its surface is able to generate large quantities of reactive oxygen species and to damage biological molecules. So this showed that the toxicity of the particles is related not only to their surface area, but also their surface reactivity. Just a few words about fibres. As most people are aware, asbestos and a number of other fibres are carcinogens. They cause cancer. And the reason that they can do this in the respiratory system and elsewhere in the body is because they're durable. That means they are biopersistent. They hang around in the body for quite a long time. And because they have a high aspect ratio, that means they are long and thin. This means that they can fly through the airways like a javelin, so that very long fibres can reach the relatively deep lung, and they're much larger than macrophages, the cells that would clear particles from that part of the lung normally. So you lead to a scenario where you have particles that are too big to be cleared from the respiratory system. And also their composition is important. For example, they can contain metals that redox cycle and generate reactive oxygen species. You put these factors together, it promotes inflammation, and this inflammation can cause disease such as fibrosis and cancer. This is a section through lung tissue with a particular type of tumour, mesothelioma, surrounding that lung tissue, because these fibres tend to accumulate in that cavity, the pleural cavity, around the lung. So we investigated asbestos and compared it to some nanomaterials that have fibre-like dimensions, such as multi-walled carbon nanotubes. So these are macrophages in culture, and you can see that they are trying to take up the asbestos fibre, but they're failing to do so. This is known as frustrated phagocytosis. They're trying to take it into the cell, they're trying to break it down, and as they try and take it in and break it down, they're generating reactive oxygen species, which can be damaging to the surrounding tissue. And you see something similar with the multi-walled carbon nanotubes, where you in fact see bundles of carbon nanotubes protruding from the cell surface. This was investigated in a mouse model by Craig Poland, who now works at the Institute of Occupational Medicine. First of all, this study involves injecting different particles into the abdominal or peritoneal cavity of a mouse and looking at the cells, mesothelial cells, that represent those cells lining the cavity around the lung. This has now been repeated in that pleural cavity around the lung, where similar results have been observed. This is the diaphragm, the muscle above the abdominal cavity, and this is coated in these mesothelial cells. This is a cross-section through that muscle with a pink layer of mesothelial cells on the surface. What you see is that in the normal health individual, it's just a single layer of cells. When treated with carbon particles, short asbestos, or short tangled nanotubes, there's very little impact on those mesothelial cells. But if you use long asbestos or long nanotubes, then you see a thickening of that layer associated with an influx of inflammatory cells or inflammation, a proliferation of those mesothelial cells, and fibrosis, suggesting that these long nanotubes have the potential to behave like pathogenic fibers. So as you can see, there's a large list of physical and chemical characteristics that can influence the toxicity of nanomaterials. And we can't possibly test all nanomaterials on a case-by-case basis in order to assess their toxicity. Instead, going forward, we need to have a more intelligent testing strategy that allows us to group nanomaterials together, to use in silico approaches, to model how nanomaterials behave in the body and in the environment. So a few years ago, we were funded by the European Commission to look at what an intelligent testing strategy might look like, and then to identify the research gaps that were preventing us from carrying out an intelligent testing strategy, and thereby developing a research strategy or a research prioritization to allow this to occur. So the project came up with a number of conclusions. It suggested that in the short term, an intelligent testing strategy would require nanomaterials to be grouped together or into classes so that you could start to extrapolate or read across from one nanomaterial to another. And to do this, you would need to understand the toxicity or the hazard of nanomaterials, shown in green here, the exposure, which is the dose, how much people are exposed to. But these are traditional in a risk assessment. But in addition to this traditional exposure and hazard information, the report suggested there would be an increased need for understanding the physical and chemical characteristics of nanomaterials, because these characteristics could help us potentially predict their toxicity and the potential for exposure. In the longer term, the project suggested there would be reduced emphasis on the need to test exposure and the need to test hazard because of improved modelling. So we'd be able to model the toxicity and model the exposure, so that in the distant future, a majority of the risk assessment would be based on modelling with just a minimal amount of testing when absolutely necessary. So I'm not going to go through all of the recommendations for the project, but for hazard, exposure and physicochemical characterization, a number of recommendations were made for research priorities that need to be considered. We need more appropriate dose metrics. Mass is not a very good dose metric for nanomaterials because they're so small, they have so little mass, and it doesn't tell you the full story. So we need more appropriate dose metrics and dosimetry studies. We need better understanding of their mode of action, because this can allow us to identify biomarkers to measure in studies. We need to be able to assess the bioavailability of nanomaterials and how they move around the body and how they move around the environment. We need standardized protocols using alternatives to animals, using animal models. We need short-term effects and long-term effects. And with those longer-term effects, we need to understand the time course of the effects. Are the effects rapid? Are they delayed? Are they reversible? Are they irreversible? For the environment, we need to understand population effects, so not just how it affects an individual, but how it can affect the survival of a population of organisms. And is there potential for biomagnification, for example, along a food chain? We need to understand susceptibility. Normal, healthy people seem to be quite resilient to air pollution, so you might expect them to be resilient to engineered nanomaterials. What makes some people susceptible? How does this influence the hazard? Where new tests are developed, they need to be high throughput if possible, so that you can test large numbers of nanomaterials, and eventually using computational or in silico approaches, so that you don't have to do too much expensive laboratory-based testing. And when, in future, we predict that a hazard can be assessed predominantly using these computational approaches rather than by testing. And we need to be able to extrapolate knowledge across different types of nanomaterials, across different types of target tissues, and even across different types of organisms. So you can actually put all of those research priorities together into a diagram that allows you to identify what the immediate things are that we can address or start to address today, and what the longer term ambitions might be in terms of understanding hazard. So things that we can do today, think about mode of action, think about dosimetry, whereas in the longer term we're developing these modelling approaches. But we need to think about what the modelling approaches are going to be like, so that we can generate the appropriate data now to allow those models to be developed. And you can generate these diagrams not just for hazard, but also for exposure and physicochemical characterization. And in ITS Nano, we put those all together to come up with a strategy for grouping and ranking, and this intelligence testing strategy. And you can see more detail on that if you're interested in particle and fiber toxicology. So one of the major conclusions from that project was that we need better models, and for the models that we have and that we're developing, we need to make sure they're relevant. Are they giving us information that's relevant to the human or animal models to allow us to do risk assessment? So in another project, we were able to look at the effects of particles on the liver, and we focused on the liver because this is an organ where nanomaterials and particles accumulate in the body. And we used in this study silver, which is a widely used nanomaterial in very many applications, and we looked at the impact on a hepatocyte or liver cell line. And we found that a number of cytokine proteins that control inflammation were upregulated in this model. Things like interleukin-8, interleukin-1 receptor, and TNF-alpha. So these are all proteins that control inflammation. And then alongside this, we had a number of animal studies. For example, these animals were injected into a vein with the same particles at a range of doses and similar or the same proteins measured. And we found that this in vitro simple hepatocyte culture was able to predict the liver responses for an injected nanomaterial exposure. Well, not many people are exposed to injected silver nanomaterials. Most people are exposed to silver nanomaterials as an oral formulation. You can buy it off the internet. It's supposed to cure you of all diseases known to man, if you believe the homeopathic literature. And in fact, if you look at them, the liver response to these silver particles was very different to the liver response for the injected particles. We did see some upregulation of TNF-alpha, but for the other cytokine proteins that we measured, there was no change. So the key is to be able to understand the model you're using and the limitations of those models. We found that simple liver culture was good at predicting the responses of injected silver, titanium dioxide, and gold particles. It was okay at predicting the liver responses to silver and titanium dioxide. It struggled with the carbon nanotubes because they probably got stuck in the lung, and didn't get to the liver. And it struggled with the zinc oxide because it was highly soluble, probably. And it was really poor at predicting the liver responses to ingested nanoparticles. If you think about the intravenous route, the particles go straight into blood, which is a plasma solution containing lots of plasma proteins or serum proteins, and our culture system contains a similar environment. We're using a cell culture medium supplemented with serum, and so it makes sense that the in vitro system can perhaps predict that intravenous injection. Whereas for inhaled particles, the particles are coated in a lung lining fluid, and for ingested particles, they're modified by the gut contents. And that leads me into two of our speakers for today. So Barbara Rawson, who is going to talk about three-dimensional lung models used for hazard assessment of nanomaterials, and Hans Baumeister has experience both in lung and gut models for investigating these particles. And the ITS Nano project also highlighted environmental impacts of nanomaterials as being a key issue to address, and a range of species are required for that kind of testing. And Teresa Fernandez, my colleague from Heriot-Watt University, is going to speak about that today too. So in the last few minutes of the presentation, I'd like to talk about 2 scenarios where we're trying to use this information now for the development and the use of nanomaterials. So it's not all about finding out why nanomaterials are hazardous. It's about using that information to help industry develop their nanotechnology in a safe and sustainable manner. So this is a project called SUN that's funded by the European Commission. It's led by Venice, and it's about sustainable nanomaterials. And we, at the beginning of the project, were presented by industry with a list of nanomaterials that they wanted to understand the hazards of, that were used in a variety of different applications. There were too many for us to test, and so we needed some way of prioritizing them. So we went through the list and we looked at the existing literature and data available for those. And actually, if you hear somebody say, oh, we know nothing about the toxicology of nanomaterials, that phrase is now outdated. There are thousands of publications in the literature on the toxicity of nanomaterials. And so we were able to cross off a couple of nanomaterials from our list, such as silver and titanium dioxide, because there was already enough information out there to allow a risk assessment to go ahead. So this allowed us to prioritize the materials for which much less data existed. So we prioritized those with lacking data. We looked at their chemistry, so anything that contained a substance that was highly toxic we prioritized. We looked at their potential for exposure and the volume of use. So are they likely to come into contact with humans, and are they being used in large quantities that are going to lead to substantial numbers of people being exposed? So we prioritized them on that basis. And we came up with a couple of priorities. Copper oxide was one of them. It's used as a wood preservative. It's incorporated into polymers and wood treatments. And so it's widely used, garden fences, telegraph poles, all kinds of applications you can imagine. And we also looked at tungsten carbide cobalt, which has more specialist application, but there's very little known about the toxicity. We then used a couple of in vitro systems, the macrophages and the liver cells, to screen this panel of nanomaterials. And we picked from those the ones that were most toxic to take forward into the next series of tests. And copper oxide came out as a highly toxic material, which is why we focused on it. Not only were we able to use that in vitro testing to prioritize the nanomaterials to test, we were also able to use it to design the subsequent animal studies, because our previous experiments in another European project that was funded before this one had used the same in vitro models with the same protocols, and the same animal models with the same protocols but with different particles. So we had a good understanding about how the in vitro dose responses related to the in vivo dose responses. So we were able to use the in vitro dose responses to precisely identify the dose range that should be studied in vivo, so that we use the minimum number of animals in order to obtain the results that we wanted. So these are the in vitro dose responses for copper oxide and some of the copper salts. And this is the in vivo dose response showing inflammation and lunging. You see we've got a good range of inflammatory response by using this in vitro data. So we showed that the particles induced toxicity in vitro and inflammation in vivo. But actually, only a small proportion of people are exposed to that pristine copper oxide nanomaterial. Most people who are going to use the wood treatment are exposed to copper oxide in a polymer matrix or in the paint. So what about the copper oxide in that? So a group in Denmark, in Copenhagen, took the polymer with the copper oxide nanoparticles in it, coated it onto a wood surface, and then they tried to grind it or drill it or sand it to release fragmented particles, and they couldn't do it. It was too sticky. They just made a big ball of sticky polymer with the particles embedded in it, showing that there was very little potential for the release of these particles or fragmented particles containing the nanomaterials to people using and handling this product. So therefore, if there's very little exposure, there should be very little risk associated with using that nanomaterial from an inhalation or an ingestion perspective. But people are still exposed to the pristine nanomaterial. Lots of people are going to be exposed in an occupational setting to the powder when it's being made and when it's being formulated. So could we modify the toxicity? So could we make it safer by design? So we worked with the chemists to come up with a number of different coatings. Here's some citrates, some different polymers, and we actually suggested ascorbate, which is an antioxidant found in the lung lining fluid, so found naturally in the body. And we measured the concentration of the particles required to kill 20% of the cell population, so increased death by 20% in the cell cultures. So you need 33 micrograms per mil of the copper oxide to induce death by 20%. If you added some of these coatings, you actually made them more toxic. But the ascorbate more than doubled the concentration that you needed to induce toxicity, suggesting that the substance is now much safer. So we're now in the process of doing an inhalation study with this to see if it's less toxic in the lung, and to assess whether the copper oxide is still useful as a wood preservative once it has that ascorbate coating. The last application for which nanomaterials that we're currently interested in using nanomaterials is for nanomedical applications. I came into contact with the Morden Research Institute in Edinburgh, who were interested in TB in cattle. And they had a mycobacterium that was genetically modified to make it fluorescently green. And this is useful because it's very difficult to handle human TB in a laboratory environment because it's so infectious, you have to have very rigorous conditions to handle it. Whereas the TB that infects ruminants or cows, you can handle it much more easily in the lab. And they had this green fluorescent protein version of that microorganism. The other problem with TB is it grows very, very slowly, so it's difficult to study in the lab. And there's a huge need for improved treatments for TB because lots of people are resistant to the antibiotics, or lots of TB organisms are resistant to the antibiotics that we would traditionally use. So we were able to use this fluorescent assay to screen different antibiotics in terms of their ability to impact on the TB organism. Normally, when you screen a TB organism with an antibiotic, it would take 7 weeks to find out if the antibiotic was efficient. But we can do this in 7 days using the fluorescent organism. So initially, we did this with a range of metal and metal oxide nanomaterials and some salts for comparison. And we found that we could, in a dose-dependent manner, inhibit the survival of this TB model in vitro. But very few people are really going to inhale silver or zinc oxide nanoparticles. So we came into contact with Andrew Owen, who's going to speak today, who is making nanoparticles from existing antibiotics. So the antibiotics already used and already regulated by the FDA for treatment. And we were able to treat the microorganism and found that they were susceptible to those particles. And we were able to infect macrophages, shown in blue here, the nucleus is blue, with the green microorganisms, shown here, and treat them with a solid drug nanoparticles such as rifampicin, shown in red. And we were found that they could co-localize them inside the macrophage. And this is because macrophages accumulate the microorganism in the same place that they accumulate particles. So we were using the particles to target the microorganism. The student involved in this work, Sam Donnellan, then lysed the macrophages to release the microorganism to see if it had survived that exposure. If she just exposed them to the aqueous drug, then about half the microorganisms survived. But if she exposed them to the solid drug nanoparticles for the antibiotic, then none of the microorganisms survived. So that's an area that we're looking to take forward in the future. So, just to summarize a few of the points that I've put forward today. There are a large range of factors that influence the toxicity of particles. The toxicity involves promoting inflammation via mechanisms such as oxidative stress. But we need intelligent testing strategies in order to better understand how nanomaterials behave and to test them in future, because we can't test them all on a case-by-case basis. And we can use the information that we do have and that we are getting to prioritize decision-making for the production and use of nanomaterials. And this information is useful not just for commercial products in paints, etc., but also for nanomedicine applications as well. I have a lot of people to thank, and I'm not going to do an Oscar-type ceremony. Instead, I'm going to say we've been lucky enough to be funded by a wide variety of organisations, many of which are listed here. And I have worked with lots of colleagues over the years across UK, across Europe. I'd particularly like to mention colleagues at Edinburgh University and the Institute of Occupational Medicine and Edinburgh and AP University who I've worked with very closely, as well as my current colleagues at Heriot-Watt University. So thank you very much.
Thank you very much, Vicky. I thought that was excellent. I almost understood most of it. Do we have any questions for Vicky? Come on. Well done.
Yeah, thank you for that talk. It was very clear, lucid, and really enjoyed it. Thank you very much. You said something about identifying dose metrics for nanomaterials. How close are you to identifying a metric that might be relevant to across a whole different nanomaterials?
The answer's not very close. It's probably going to end up being some kind of algorithm, maybe of multiple physical and chemical characteristics. I think maybe for some classes of nanomaterials, you might end up with something fairly simple, like surface area for low toxicity, low solubility materials. But for materials that are more complex, it's probably going to end up being a combination of factors. So rather than having a dose metric, you might end up with some kind of QSAR, quantitative structure-activity relationship instead. So there's nothing clearly coming out at the moment that's going to help us say this one thing is going to solve our problem.
And just leading on from that, as toxicologists, we tend to try and think of threshold and non-threshold responses. So can you see a threshold dose for nanomaterials once you've got your dose metric identified?
So people are tending to use things like LC20s or BMD20s, so the lethal concentration that increases toxicity by 20% or a benchmark dose for 20%, rather than a threshold. I think as the modelling approaches improve, maybe you can get lowest observed effect concentrations, for example, in environmental scenarios. But I wouldn't say thresholds per se have been identified. There was some work about 10, 15 years ago which tried to look at surface area thresholds, and Lang Tran and Ken Donaldson and a few others came up with a surface area of 1 centimetre squared per centimetre squared of lung surface area as a threshold, but that's never really been pursued any further.
Thanks, Vicky. That was really nice. I was intrigued by the use of ascorbate then, and quite a dramatic shift in the potential toxicity. So, is mechanistically then, what do you think's happening?
Well, ascorbate is an antioxidant that's used naturally in the body. So I think is
it an ROS? Is it just like mopping up, helping to prevent?
That would be my hypothesis,
ROS,
but we haven't been able to test that yet.
Right.
What I want to make sure is that ascorbate is truly associated with the particle surface and not just a background in the media in which the nanomaterials have been made. One of the issues that we often have is that when we're handed a nanomaterial, we ask certain things about the characteristics and we're told them, and then we ask, how do you know that? And the answer is, it's because that's what we put in the tube, not because that's what we measured. So we need to go back and make sure that ascorbate really is coated on the surface of the particles and it's not just free antioxidant in the media, because we already know from our experiments from 18, 20 years ago that if you add an antioxidant into the cell culture medium or into the animal, then you can protect against the pro-inflammatory effects of nanomaterials.
Yeah, so I guess that's the question, is whether or not it could be used then in the production and added in.
Yeah, I mean,
maintained.
Even if it doesn't need to be coated onto the particle surface, even if you could add it into the product, that might help in some way. Teresa can ask me tomorrow at work.
It's lovely lecture. Thank you very much. That's really inspiring. I wanted to ask you, we understand very well now that the fiber-induced pathology, on the example of asbestos and mesothelioma, can have a very long period for development. So the latency for mesothelioma can be up to 40 years, as we know. So how this knowledge is integrated into your intelligent testing of materials?
So I think this is a really key question, and that nobody has a good answer to at the moment. And without divulging all of my recent ideas, quite often we measure acute pro-inflammatory effects, but we very rarely measure the ability of that inflammation to resolve or to be sustained. And I think that in order to address these longer term effects, we need, if we believe inflammation is key in driving those longer term effects, we need to not just look at the acute effects, we need to look at the ability to resolve as well. And there's nothing, or hardly anything on that in the literature. There's a short-term inhalation study model now that's been proposed as an alternative, and is being used as an alternative to the 90-day inhalation study that the OECD recommends and that many regulators require. And that short-term inhalation study requires you to expose animals for 5 to 7 days, and then you stop the exposure. And after you stop the exposure, you measure the inflammatory response then, and you measure it again at day 28. And that does give you some information then about the ability of that inflammation to resolve. But that's really the first data, and that requires an animal, and it requires a 28-day study. So are there alternative ways of addressing that is something that I'm really keen to investigate.
Super presentation, Vicky. Thank you very much. So I have a question. As a pharmacologist, when you think about immune-mediated adverse drug reactions, they tend to be non-dose dependent or not concentration dependent reactions. Is there any evidence for that across the nanomaterials?
Most of the immunological endpoints that we've measured, such as accumulation of neutrophils or accumulation of macrophages or pro-inflammatory cytokines, are dose dependent, but you need to be careful about the time that you look at, time points. So they're highly time dependent as well. So sometimes what happens is, instead of getting a nice dose dependent sigmoid curve, you get a bell-shaped curve. And the reason being, it depends on the dose. If you use a really high doses down to low doses, your high doses are so toxic to the cells that they can't promote inflammation, they can't make pro-inflammatory signals, and whereas the lower doses can. So it becomes confusing. And also, it's very complicated. I think you need to think about the dose range and the time range. So I don't know very much about the pharmacology literature, but it'd be worth looking at it with that in mind, perhaps. One suggestion.